This is Jahangir from MedPorium, and today we'll talk about the urinary system, specifically part 2 of the urinary system. In part 2, we'll discuss the first step of urine formation, which is glomerular filtration. So let's answer the question, what is glomerular filtration? Well, glomerular filtration is a process in which water and solutes of the blood plasma pass from the capillaries of the glomerulus into the glomerular capsule. Let's move over to the ThinkPad to understand what glomerular filtration is. To understand glomerular filtration, we also want to understand the mechanisms of urine formation. We'll start by glomerular filtration. So glomerular filtration creates plasma-filtered fluid from the blood which moves on over through the renal tubule. Second, we have tubular reabsorption, which is a process of releasing useful solutes from the fluid and return it back into the blood. An example would be salt. Next, we have tubular secretion, which is the process of removing additional waste from the blood and adding it into the filtrate, making the urine more concentrated. Finally, we have water conservation. The tubules reabsorb water from the urine and add it back into the blood, making urine even more concentrated. So now let's talk about the 3 different names that reflect composition throughout the nephron. We'll start from the capillary space. Fluid in the capillary space is referred to as glomerular filtrate, which is similar to blood plasma but without the protein, since protein cannot pass through the filtration membrane. We'll talk about the filtration membrane in the next slide. Fluid from the proximal convoluted tubule all the way through the distal convoluted tubule is referred to as tubular fluid, and fluid is referred to as urine once it enters a collecting duct. Now let's talk about filtration membrane. What is a filtration membrane? Well, a filtration membrane is a barrier in which solutes and water of the blood plasma pass through to enter the capillary space of the nephron. There are 3 barriers of the filtration membrane. The fenestrated endothelium of the capillary, which is highly permeable with pores that exclude red blood cells and allow passage for small solutes. There is the basement membrane protein, which has a protoglycan gel which stops large molecules from passing through the membrane, and filtration slits, which are areas between each pedicel, which are extensions of podocytes, also called foot processes. Filtration slits are negatively charged and prevent large anions from entering. Now let's move over to the ThinkPad to really understand what the filtration membrane is. So this picture represents the anatomy of the filtration membrane. As we talked about from the 3 barriers of the filtration membrane, we have our fenestrated endothelial capillary, we have our glomerular basement membrane, and we have our filtration slit. The filtration slit are the space between each foot process of the podocytes. Now let's talk about what can and can't pass the filtration membrane. What can't pass the filtration membrane include blood cells, plasma proteins, large anions, just basically molecules bigger than 8 nanometers in diameter. Now let's talk about what can pass through the filter. What can pass through the filter include water, electrolytes, glucose, amino acids, nitrogenous waste, etc. Just basically anything less than 8 nanometers in diameter. Now there are some solutes that are bound to plasma proteins, which means they don't filter. So let's just say this is our plasma protein and this is our solute. Now this compound or the solute doesn't get filtrated because it's bound to the plasma protein, and as we talked about before, the plasma protein does not pass through the filtration membrane. Now, kidney infection and trauma can damage the filtration membrane, allowing these plasma proteins and, let's just say this is our red blood cell, and red blood cells to pass through the filtration membrane and into the urine. The condition in which plasma proteins are found in the urine is called proteinuria or albuminuria. Condition in which blood is found in the urine is called hematuria. Now let's talk about filtration pressure. To understand filtration pressure, let's move over to the ThinkPad. There are three coefficients that account for filtration pressure. The first one includes blood hydrostatic pressure, which is normally 60 millimeters of mercury. It's 60 millimeters of mercury because the afferent arteriole is larger than the efferent arteriole, which just basically means that there's a larger inlet than an outlet. It's normally 60 to 55 millimeters of mercury. Next we have colloid osmotic pressure, which is usually 32 millimeters of mercury. And this is just resistance that the filtrate must overcome. The 60 millimeters of mercury must overcome. Now that's the same for capillary pressure, but instead it is 18 millimeters of mercury. And this is just normal numbers that you'd see in a normal patient. Now this is just resistance, just like I said, for the colloid osmotic pressure. And this resistance is due to high rate of filtration and fluid accumulation. Now to find net filtration pressure, we just subtract blood hydrostatic pressure with colloid osmotic pressure and capillary pressure, which usually is 10 millimeters of mercury for a healthy patient. So that is the basics of filtration pressure. Now let's talk about glomerular filtration rate. Now what is glomerular filtration rate? Well, I'm glad you asked. Glomerular filtration rate is the amount of filtrate formed per minute by both kidneys, and this shows kidney function. The filtration coefficient, Kf, shows membrane permeability to water and surface area of a filtration membrane. Now let's move over to the ThinkPad to really understand what glomerular filtration rate is. To understand glomerular filtration rate, we need to know how to find glomerular filtration rate. So glomerular filtration rate is found by identifying the net filtration pressure and multiplying that by the filtration coefficient. Now, as we talked before, the normal net filtration pressure of an adult male is usually 10 millimeters of mercury. Now, for the filtration pressure, a healthy adult male produces 12.5 milliliters per minute of filtrate for every 1 millimeter of mercury from the net filtration pressure. So then that would amount to 10 times 12.5, which would equal to 125 milliliters per minute, which is the normal glomerular filtration rate of a healthy adult male. For women, it would be about 105 milliliters per minute. So let's talk about regulation of glomerular filtration. Now, regulation of glomerular filtration must be precisely controlled. If glomerular filtration rate is too high, fluid rapidly moves through the renal tubules without reabsorbing needed water and solutes to go back into blood, which can result to dehydration and electrolyte depletion. Now, if the glomerular filtration rate is too low, fluid moves slowly, reabsorbing waste that should be eliminated, which equates to azotemia. Regulation of glomerular filtration is achieved by three homeostatic mechanisms. The first is renal autoregulation. And this is the ability of nephrons to adjust their own blood flow and glomerular filtration rate by themselves, which helps ensure stable fluid and electrolyte balance in spite of external situations, such as high blood pressure. There are two mechanisms of renal autoregulation. The first includes myogenic mechanism, which is a mechanism of stabilizing blood flow and glomerular filtration rate based on changes in blood pressure. For example, when arterial blood pressure is high, it stretches the afferent arteriole, and the afferent arteriole reacts by constricting itself, which keeps the blood flow constant. The second mechanism of autoregulation includes the tubulo-glomerular feedback mechanism, and this is a mechanism in which the glomerulus gets feedback status from downstream tubular fluid and adjusts filtration to regulate composition, stabilize nephron performance, and deal with fluctuations in blood pressure based on that feedback. And this feedback is mostly based on salt and other ion concentrations in the macula densa. We'll talk about the macula densa later. The tubuloglomerular feedback includes the juxtaglomerular apparatus. Let's move over to the ThinkPad to understand the juxtaglomerular apparatus. So we're at the ThinkPad and we want to understand what the juxtaglomerular apparatus is. Well, the juxtaglomerular apparatus is a structure at the vascular pole of the renal corpuscle that maintains blood pressure through three special cell types. The first is the macula densa, which are patches of closely spaced epithelial cells at the end of the nephron loop. Second, we have the juxtaglomerular cells, or granular cells, which dilate or constrict the arterioles with granules of renin, and they secrete this in response to drop in blood pressure. We'll talk about what renin is later. Third, we have the mesangial cells, which are connected to the macula densa and the juxtaglomerular cells. They communicate or talk to each other by paracrine secretion or paracrine secretion. They build supportive matrix for the glomerulus, regulate blood flow and glomerular filtration rate by constricting or relaxing the capillaries, and they phagocytize tissue debris. Now, to understand the juxtaglomerular apparatus, let's move over to the next slide in the ThinkPad. Alright, so we're at the next slide of the ThinkPad and we want to understand the juxtaglomerular apparatus and how it functions. So let's just say we have a high glomerular filtration rate. This high glomerular filtration rate means that we have a high salt reabsorption by the proximal convoluted tubule. Now this high salt reabsorption will be detected by the macula densa, and the macula densa will secrete a paracrine messenger to the granular cells or the juxtaglomerular cells and tell them, hey, can you constrict the afferent arteriole for me since there's a high glomerular filtration rate? So then the granular cells say, no problem. So they constrict the afferent arteriole, leading to a low glomerular filtration rate, which means a lower salt reabsorption by the proximal convoluted tubule, which kind of restores the balance. The mesangial cells also help by constricting the glomerular capillaries, which means a reduced filtration, which, as we talked about, equates to a low glomerular filtration rate. Now, this is the same if there's a low glomerular filtration rate, and the opposite occurs. If there's low glomerular filtration rate, the macula densa secretes a paracrine messenger telling the granular cells, hey, can you relax the afferent arteriole? Granular cells say, yeah, no problem. So they relax the afferent arteriole, leading to a high glomerular filtration rate, and then it would equate to a higher salt reabsorption by the proximal convoluted tubule, restoring homeostasis. So we just wrapped up our conversation from the ThinkPad about the juxtaglomerular apparatus. Now let's talk about the second homeostatic mechanism of the kidneys, which is the sympathetic control. Now, sympathetic control refers to the sympathetic nervous system, which directs the body's rapid involuntary response to dangerous or stressful situations. In vigorous exercise or acute conditions such as circulatory shock, sympathetic stimulations and adrenal epinephrine constricts the afferent arterioles, which redirects blood where it's most needed, such as the brain or the heart. This equates to low glomerular filtration rate and low urine output, followed by low perfusion. Now, the sympathetic control is the last resort, giving necessary oxygenated blood to the organs that need them, such as the brain or the heart, or if you're a distance runner and you're running, to the muscles that need it. So now let's move on to the third homeostatic mechanism that the kidney has, and this is the Renin-Angiotensin-Aldosterone Mechanism. Let's go over to the ThinkPad to really understand the Renin-Angiotensin-Aldosterone Mechanism. To understand the Renin-Angiotensin-Aldosterone Mechanism, we want to ask the question of when is this mechanism activated? Well, the Renin-Angiotensin-Aldosterone Mechanism is activated when there is a drop in blood pressure, in which the sympathetic fibers stimulate the juxtaglomerular cells to secrete renin, as I talked about before. Now, renin breaks down angiotensinogen, which is produced by liver, into angiotensin 1. Now, the angiotensin converting enzyme breaks down angiotensin 1 to angiotensin 2. Angiotensin converting enzyme, or ACE, is produced by the lungs. Now, angiotensin 2 has many different impacts over the body. We'll start by the adrenal cortex. Angiotensin 2 stimulates the adrenal cortex to release aldosterone, and aldosterone is claimed as the salt-retaining hormone. Aldosterone increases sodium and water retention, which overall elevates blood pressure. Angiotensin 2 has a vasoconstriction effect on the efferent arteriole more than the afferent arteriole, which means a larger inlet than an outlet. As we talked about, from a larger inlet than an outlet, this increases glomerular filtration rate and it increases glomerular blood pressure, which ensures continued filtration of waste from the blood. The increased glomerular filtration rate and the increased glomerular blood pressure would eventually equate to an elevated blood pressure overall. Now, angiotensin 2 also has an impact on the brain, as it stimulates the posterior pituitary gland to secrete antidiuretic hormone, or ADH, which increases water absorption and overall elevates blood pressure. The whole purpose of the renin-angiotensin-aldosterone mechanism is to increase blood pressure when it's low. Alright, so that is going to wrap up part 2 of the urinary system. In part 2, we discussed the first step of urine formation, which is glomerular filtration. We also talked about the filtration membrane of the glomerulus, the filtration pressure of the glomerulus, the glomerular filtration rate, and the regulation of glomerular filtration. And as always, a Pomodoro a day makes you Dr. Pig. I'll see you next time.